Well, thanks very much, Ed, for that kind introduction, and thanks to Jennifer for inviting me to give this founders lecture. Actually, I wouldn't be standing here this morning if it weren't for Ed and Jennifer back in 1986. So how do we get the first slides? So I'm going to give a kind of historical overview, or a trip down memory lane, because 30 years ago, two very significant scientific developments were published, and they really revolutionized the field of forensic genetics. So in 1985, Alec Jeffreys and his colleagues at Leicester University in the UK published a paper on that they called DNA fingerprinting. So it was generating these complex patterns that you can see with P32 labeled probes using a technique known as the Southern blot after Ed Southern. These probes hybridized to regions of the genome that had variable length. So they were known as VNTRs, variable number tandem repeat. There's a length polymorphism, and you can see that it creates a very complex pattern. And Jeffrey's contribution was to point out that this pattern could be used to distinguish human individuals and to do individual identification. As Ed mentioned, the same year, our paper on PCR was published, and as Ed explained, this was a method for the enzymatic amplification using DNA polymerase and targeted oligonucleotide primers to synthesize millions and millions of copies of a specific targeted gene. So DNA fingerprinting was very powerful. It used a Southern blot, a technique known as restriction fragment length polymorphism. They required a lot of DNA, and the DNA had to be intact. That is, if it were degraded, this approach wouldn't work. And as I said, the initial probes for VNTR loci were P32 labeled, but they hybridized to many, many different loci. So you had a very complex pattern. It was difficult. The criteria for establishing a match or an inclusion between the forensic specimen and the reference sample were not clear. And if you did decide it was a match, it wasn't clear what the significance was, because there were no statistical databases, so that although very powerful, it had a lot of limitations. Now, these limitations were addressed in the subsequent development of multiple single locus probes, and that meant one could develop population databases for these single VNTR loci. You could therefore make statistical estimates for inclusions or matches. And because these loci were assumed to be statistically independent, one could use the product rule and generate very powerful random probability estimates, random match probability estimates. So in the US, the first RFLP case was the Tommy Lee Andrews case in Florida, which was a rape, and resulted in his conviction based on RFLP data. But the first challenge to this, and this turned out to be very significant, was People versus Castro in New York in 1989. A very distinguished scientist, Rich Roberts, was an expert witness for the prosecution. Another distinguished scientist, Eric Lander, was a witness for the defense. And in the ensuing discussions, it was clear that some of the criteria for calling a match were not very rigorously defined. And the judge ruled in this case that exclusionary results could be presented to the jury, but that an inclusionary result was inadmissible. Now, all of these first cases were done by commercial labs, and as a result of the discussion in People versus Castro, the FBI got involved, and I think that was a very good thing for the field because it resulted in standardization. And you're all familiar now with twig dam guidelines, and that was partly a result of the discussion and controversy surrounding People versus Castro. Now, as I said, in 1985 we published our first PCR paper, and it described this exponential amplification of the targeted gene. And as Ed indicated, it is very, very powerful, in particular in forensics, although it's been used in all areas of molecular biology. Because we can synthesize millions of copies of a targeted gene, we could then analyze minute quantities of DNA, so in a hair shaft, cigarette butt, blood drop, and also we could analyze degraded DNA, which, as you know, is often the case for forensic specimens, certainly in missing person studies with human remains. The other very useful thing about PCR is really just an enabling technology. The amplified DNA can be analyzed by determining the sequence, by hybridizing what's labeled oligonucleotide probes, and in the case of length polymorphisms, by simply running the amplicons on a gel or capillary to genotype them based on a length polymorphism. And because it provided specific genotypes with well-defined alleles, this was a more robust system than the original RFLP loci, many of whom had literally hundreds and hundreds of alleles, and so estimates of frequency were not very robust. So one of the first significant impacts in the forensics community when we developed PCR, and actually because Ed was down the hall, we got involved in forensics. One of the first contributions was analysis of single hairs. And this paper we published in Nature. Russ Iguchi was the first author, and we analyzed the polymorphic gene HLA, which I'll talk about, and then we also sequenced the mitochondrial DNA from these hairs. And one of the things that was immediately clear, and I think quite disturbing to the forensic community, was that the genotyping of hair revealed the subjective and error-prone nature of hair morphology analysis. So the number of hairs that had been judged to come from the same person could actually be shown to be from different people. And when we first published PCR, we amplified the beta globin gene because our interest was in clinical diagnosis. So the first papers involved a prenatal diagnosis of sickle cell anemia, amplifying and genotyping the beta globin gene. But because of my long-standing interest in immunogenetics, the second gene we amplified was an HLA locus, and these are very, very polymorphic genes. And so we then were interested in the potential of applying this system to forensics. So, as I mentioned, HLA is very polymorphic, so it's very useful for population genetics, for forensics, for paternity. But I was also a researcher interested in clinical translational projects, and as you may know, matching the HLA genotype for the donor and recipient in both hematopoietic stem cell transplants and solid organ transplants was crucial. So there was a clinical application for developing an HLA DNA typing system, and there are a large number of diseases where a specific allele at one or many of the HLA loci is associated either with greatly increased susceptibility or increased resistance to autoimmune diseases, cancer, infectious pathogens. We've done a lot of work on the host genetics involved in HIV, and also in pharmacogenetics. We developed a test for Bacavir, which is antiretroviral drug used to treat HIV patients, and about 5% of all patients treated with that have a severe allergic response, and that's 100% associated with a particular HLA allele. And as some of you know, the HLA genes are the most polymorphic in the human genome. So when we first started doing HLA DNA typing, people said, well, why bother? We have a serologic system for HLA typing and matching donor and recipients. And for instance, at the HLA-B locus, there are 68 distinguishable serologic specificities, but we now, and this is a couple of years out of date, there are many, many thousands of alleles at these loci. So these are by far the most polymorphic genes in the human genome. And the reason is because all of the polymorphism is localized to the part of the peptide binding groove that binds a peptide, and this is how the immune response is initiated. When the T cell receptor on the T lymphocyte recognizes a specific peptide in a specific HLA molecule. And that's why HLA genes are polymorphic for fundamentally different reasons than SDRs or VNTRs. That these genes are polymorphic because natural selection wants to maintain as many variants in the population as possible so as to increase the likelihood that people can respond to different pathogens. Whereas, as you know, like polymorphisms are polymorphic because they're mutable. Now, interpreting the significance of a match obviously requires knowledge of the frequency of that genotype in various population databases, and that's how we, thank you. That's how we determine the significance of a match, or the random match probability. But the same data, the absolutely same data, can be used to study human evolution. And here is just a phylogenetic tree based on HLA, allele, and haplotype frequencies for just two different loci. So here you have sub-Saharan Africa, here you have the Middle East, here you have Europe, and then it spreads out for the rest of the world. So it's, the data, I mean, what's very interesting about genetic population data bases are that they can be used to reconstruct the immediate past, the crime scene, but they can also be used to reconstruct the ancient past by looking at the migration and evolution of different human populations. So this was actually the first commercial PCR test launched, HLA-DQ alpha test. And it was certainly the first forensics DNA kit. And here, this is an immobilized probe technology for short oligonucleotide probe is immobilized on this membrane. And then when you're amplifying the polymorphic gene, you can label it during PCR and then hybridize it. So this is just a case where this is obviously an exclusion, where the sperm fraction does not match the suspect, but the epithelial cell fraction, as you might expect, matches the victim. And this was a system based on incorporating a variety of different SNPs, bi and tri-allelic loci, into the same multiplex PCR. So all of this was really made possible through the introduction of a thermostable DNA polymerase. This was isolated by my colleagues David Gelfand and Suzanne Stoffel. And the Taq polymerase was isolated from a thermophilic bacteria in Yellowstone. And when we introduced this heat-stable DNA polymerase into PCR, it made it more specific, more efficient, and equally importantly, more automatable with simple thermal cycling instruments. Before, when we were using an E. coli DNA polymerase, every time you heated the reaction to separate the two strands and denature the double helix, you inactivated the enzyme, and so you had to open the tube and add more enzyme back. With the heat-stable DNA polymerase, you could just add everything in the tube and just cycle the temperature in the tube. And so that's really what made PCR a robust and now universally used system. This actually shows you that what introducing a thermostable polymerase as opposed to an E. coli DNA polymerase did for PCR. So this is an ethidium bromide stained gel at 20, 25, 30, 35 cycles. And this is 35 PCR cycles of genomic DNA from a cell line that's deleted for beta globin. So just looking at this, you have no idea at all that you're amplifying anything. You're presumably have primers for beta globin. You're hoping to amplify beta globin, but when you look at what was synthesized in the tube, it's just a smear And there's no difference between a genome that has beta globin and a genome that doesn't on a histidine bromide stained gel. But if you do a southern blot with P32 labeled probe, you can see, oh, we actually did amplify beta globin. It's absent in deleted controls, but about only 1% of everything we synthesized in the tube was beta globin. But with TAC polymerase, you can see that it is very specific and very efficient, and as I said earlier, automatable. So the very first case where we ever applied PCR to human identity was actually turned out to be the first case in the United States, Pennsylvania versus Pestanikas, and it was really because I'd run into Ed in the hall, and we would discuss genetics and the possibility that DNA might be useful someday in forensics. And so at one point...
Did we ever talk about going up Highway 1?
No, we forgot.
Yeah, this that one.
Yeah, we didn't get a chance to talk about tablecloth either. Yeah, that was puzzling introductions. I never realized the tablecloths were so critical to forensic genetics. But at any rate, the Pestanikas case was challenging for Ed because these were samples that had been stored in formaldehyde. They were autopsy samples, and so the proteins were denatured, and so that eliminated the possibility of analysis by protein electrophoresis and serology, which was, those were what we had in our arsenal at that time. And it was an interesting case because the prosecution claimed it was negligent homicide by a nursing home, and their hypothesis was that these autopsy samples from the victim had been switched by the nursing home. So their hypothesis was that if one did a genetic test, the autopsy samples would not, in fact, match the reference sample from the victim. So that was the challenge. And at first I said, since this is 1986, I'm not sure what we could do. I'm happy to try, because I was always very interested in the possibility of using some of the DNA technology in practical settings like forensics. I'd always been interested in forensics. So I said, well, we can try southern blot and do RFLP, because we had non-radioactive probes for looking at polymorphic loci. But when we looked at the DNA, it was degraded into very, very small fragments by the formaldehyde. So there's no way that an RFLP analysis was possible. In fact, in the lab, we joked that the DNA was so degraded it was more like nucleotides with pretensions. But anyway, I told Ed that we were working on this new system, PCR, and we just started using the TAC polymerase, and we had some experience amplifying polymorphic locus, and we had experience with oligonucleotide probes genotyping the various alleles at this locus. So I said, we'll try it, and George Bugawan, the technician in the lab, got it to work. It worked well. The result was the D2 alpha genotype of the samples actually matched the genotype of the victim's reference. So this was a kind of interesting case where we had been asked by the prosecution to carry out the test, but the results favored the defense, and therefore the results were not challenged and went straight into the courtroom. And so in my naive ignorance, I thought, oh, this is how new technology enters the judicial system. Some scientist comes up with something and it goes right in. Well, many, many years later, I realized how naive that idea was after sitting through many, many admissibility hearings. And I'm going to discuss some of those issues in a minute. So this was the team that did the original forensics case. There's Ed, who brought us the samples, and George Bugawan, my technician, who actually did the analysis. Russ Iguchi didn't work on this project, but he was the one, as you may recall, who developed an analysis of hair. And then there are two people who just wandered by when this picture was being taken. But what we found, as we went more and more cases, was the nature of the adversarial system. That every time there was a inclusionary result, the defense would say, oh, this is really kind of junk science, and we don't really believe this, and it shouldn't be presented to the jury, it should be ruled inadmissible. And when there was an exclusion, that often the prosecution would argue that this was unreliable and should not be admitted. So that was our introduction, or my introduction, to an adversarial judicial system. So with RFLP admissibility, one of the approaches was to allow exclusionary DNA results to be presented to the jury, as in the People versus Castro and also Minnesota Supreme Court decision, but to not allow inclusion or matches to be presented. In other words, the idea was include exclusion and exclude inclusion. So that was not totally unreasonable, but it really didn't allow what we now know is the immense power of DNA analysis in the course of justice. That is, if you're excluding inclusions and matches, there are many people who are guilty who may not be convicted because you've excluded very critical data. Now these the DNA wars were mainly about statistical estimates of population frequency, and this was a debate that went on in the scientific literature by very distinguished scientists. So one of the first shots across the bow, as it were, was his paper DNA Fingerprinting on Trial by Erlich, in which he argued that, at least for RFLP, the criterion for determining a match were not robust, and that even when you did the way to estimate the random probability, random match probability was unreliable. And this was countered by two other very distinguished forensic scientists, Chakraborty and Ken Kidd, who argued that in fact the statistics were reliable, and Lewinton and Hartle responded, again, mainly a critique of population genetic statistics, and Bruce Weir, another very distinguished population geneticist, responded. But finally, there's a paper written by Eric Lander and Bruce Bedoli, Eric being a critic of DNA fingerprinting, and Bruce, who worked at the FBI, being a supporter. Presumably this issue was laid to rest. That was laid to rest partly by making statistical assumptions which were very much overly conservative, but the critical point was that in admissibility hearings now there was a feeling that the scientific community had reached a consensus. But in a way, these DNA wars, which were all about RFLP, were kind of irrelevant. I mean, it's ironic that most of the critiques and controversy that addressed issues with RFLP were happening just as the forensic community was shifting away from RFLP because of all the technical issues to PCR-based genotyping. In fact, one of the strong arguments by Eric Lander and others that our assumptions for calculating random match probabilities was invalid because they said there was a deviation from Hardy-Weinberg equilibrium proportions in many databases. So, for some of you who may not remember this, Hardy-Weinberg equilibrium is a way of predicting genotype frequencies based on observed allele frequencies given certain assumptions. And so it's actually a very robust system. But in some cases in these databases, there was an excess of homozygotes that was observed versus what would be expected under Hardy-Weinberg equilibrium. So in fact, you're observing too many homozygotes. So Eric Lander and others said, oh, that means there's population substructure. A lot of the populations that we think are relatively homogeneous are really substructures, and we can't uses statistical estimation systems that we have been using, but it turned out Neil Rich and Bernie Devlin showed that in fact the excess of homozygotes really had nothing to do with population substructure. It was a technical problem with RFLP technology, that you might have a heterozygote where if you had a really good gel, the two variants would be separated. It was a heterozygote. But if they ran together, you would incorrectly score that as a homozygote. So I think my general point is that a lot of this controversy was irrelevant because it was directed toward RFLP data, which was on its way out. So my experience after the Pennsylvania versus Pestanikas in admissibility hearings was actually very instructive to me. And I'm just going to go through a few of these cases because they illustrate some general points. So the first case where I testified personally was in California versus Mack. So Mack was accused of rape and murder. There was a match, that is the sperm fraction and the semen stain matched Mack. And so in the admissibility hearing, I was going over the nature of the data. And apparently my testimony convinced Mack, but it didn't convince the judge. Because Mack changed his testimony. Originally he said he didn't know the victim, and after my testimony he said, well, you know, I did know her, we did have sex, but it was consensual. You know, I did not rape and murder. But the judge decided this new technology, since it wasn't being used yet by the FBI, should not be admitted to the jury. So as a result, the jury never saw the data, but Mack did change his testimony. He was ultimately convicted of both rape and murder. Shortly after Pennsylvania versus Pestanikas, we did the first post-conviction review, where someone who had been in jail for about 10 years, over 10 years, for rape should be shown to be innocent. And the most dramatic case I was ever involved in was Virginia versus Washington, which I'll talk about in a minute. But I now want to discuss briefly a case called California versus Mello, where there were two suspects, Mello and Modesto, who had been accused of rape and murder. And a discussion of this case, I think, raises the question, is an adversarial system the best way to evaluate new technology and determine admissibility? And my takeaway from this is not so much. Anyway, as I said, Mello and Modesto were suspected of rape and murder. The state had made a deal with Modesto, who was convicted of murder. He was underage, so Mello, because had the prosecution been able to convict Mello of rape and murder, they were then going for the death penalty, because he would be eligible for a death penalty if he were convicted of both rape and murder. Modesto was a minor, so he was not eligible for the death penalty. So they made a deal with him and convicted him of murder, and then went on to prosecute Mello for both rape and murder. The problem there was that the DQ alpha result excluded Mello. So this is the victim genotype, this is Mello's genotype, and perhaps one reason that they're related, that they're similar, is that the victim was Mello's grandmother. But anyway, you can see that the sperm fraction was a 2-4, which is an exclusion of Mello. So the prosecution tried to exclude this from being presented to the jury. Ultimately, the judge decided it should be presented to the jury, and Mello was convicted not of rape, but of murder, and so didn't get the death penalty. But as you can see here, a somewhat awkward result for the prosecution. The sperm fraction matched the guy they made the deal with. And actually, if I can come back to this system, in other judicial systems, the expert witnesses do not testify for one side or the other. They testify for the court. Basically, they can advise the court whether this technology is reliable and is appropriate to be presented to the jury. Whereas in the adversarial system, there's a huge incentive, as you can imagine, to object, because who's paying your bill?